Well, we are continuing with biomolecules, and now we're going to be talking about my favorite biomolecule, which is the protein. But before we do, let me ask you, how many of the 5 words from the very first day of class do you remember? Do you remember those 5 words? Do you remember me telling you a story about a boy and a man? That was a hint. All right. Probably you remember all of them, right? You remember fish because the granddad took his grandson fishing and they put the fish in a red wagon and grandfather was in there and they tied the wagon to a kite and pulled it to a church, right? Now, you haven't been trying to remember that, but you remember it. And that shows you that the human mind loves stories. When you turn these physiology concepts into stories, and they really are all stories, then you will remember them too. Not just for now, but when you need them in your next program, in your clinical program. So far, we have talked about carbohydrates, and we just finished talking about lipids, including phospholipids. So now we're going to talk about proteins. Some proteins are what we call structural, like the protein that your fingernails are made out of, that's structural. Proteins that your hair is made out of, or that your tendons are made out of, those are structural. But I would go out on the limb and say that most proteins are actually like robots. Now, they're not like fun robots like this, right? They're not like C3PO and R2D2. They're more like assembly line robots where they grab something and do it and grab and do it over and over and over again. And so proteins, whenever we say that this organ of the body does something, like if I say that the liver eliminates alcohol from your blood, it's not the liver that's doing it. It's cells in the liver that are doing it. It's not even cells in the liver that are doing it. It's a protein that's doing it. Almost all the stuff that gets done to create a living cell gets done by proteins. And yeah, a lot of proteins are like assembly line robots, either putting things together or ripping things apart. Some proteins are like conveyor belts. Some proteins are like revolving doors. We're going to be talking about proteins. Now, proteins are also called polypeptides. If someone has got a reason for you to know a distinction between a polypeptide and a protein, fine, but that won't be me. As a matter of fact, protein biochemistry was my field, and I'll tell you honestly that molecules known as peptides are actually just really short proteins. So a peptide is actually just a really short protein. So whether we're talking about a peptide or a polypeptide or a protein, I don't know where it went. There we go. Peptide or a polypeptide or a protein, they are polymers made out of monomers that are known as amino acids. And they really are a chain of amino acid. So in a sense, building a protein is like stringing beads onto a strand that will become a necklace. In these strands that become necklace, there are 20 different beads. So instead of just like black, white, red, yellow beads, there are 20 different beads. And they can be combined in an endless number of ways to create these molecules that act as robots that are called proteins. All righty? Now, these are the 20 different amino acids. I don't need you to memorize them. I'm not even going to ask you to memorize the names or the abbreviations for the 20 different amino acids. The reason that I'm showing you these pictures is so that I can actually reveal to you that the amino acids are not different in their general structure. In their general structure, every amino acid has got an amino group on one side that is attached to a carbon atom. And then there's a hydrogen in the second place where you can have a covalent bond on a carbon. And in the third place, for a covalent bond on a carbon, there is a carboxyl group. An amino group is NH2 or NH3. This is chemistry for biologists. A carboxyl group is COOH, right? Now, all of the amino acids have got an amino group, a carbon, a hydrogen there, a carboxyl group, all of them. And all of those 20 also have what is known as a side group. They're the same except for there are 20 different side groups. It is the side group that is variable. So let's go back to those side groups. Now, the side groups in this image are boxed out in white. So I'd like you to notice that this particular amino acid glycine, its side group is just a hydrogen? Well, that's crazy, right? Okay, just a hydrogen. Glycine is special because its side group is so tiny. Now, completely on the opposite end of the spectrum, let's look at tryptophan. Tryptophan has an enormous side group. It's got this CH2, and then it's got this pentagon kind of structure, and then it's got this hexagon structure, which is an aromatic ring. Wow, that is really, really big as a side group. So imagine in your mind's eye that you're making a necklace, and one of those beads is a glycine, and it's got a teeny little hydrogen hanging off of it, and another of those beads is a tryptophan, and it's got this big old thing hanging off of it, right? So we're putting beads on the chain, and those side groups are different. Now, not only are there small side groups and big side groups, but those side groups will end up interacting with each other. They'll interact with each other. Why? Because of some of the concepts we've just been discussing. We've been discussing polar and non-polar, hydrophobic, hydrophilic, right? We've been discussing ionic bonds. We've been discussing charge and ions, right? All of those things are ways that these side groups can be different from each other. I'd like you to imagine in your mind's eye that you are going to make a string of amino acids as a protein, and you're going to put an arginine, which has got a charged side chain with a positive charge, and next to it, you're going to put an aspartic acid, which has got a negative charge, and then you're going to put another arginine, and then another aspartic acid, and another arginine, right? Give it a little bit of thought. I know I said arginine and aspartic acid a bunch of times, but what you did was you had a positively charged side group, a negatively charged one, positive, negative, positive, right? Now, in your mind's eye, think about positive, negative, positive. How do oppositely charged ions interact with each other? They click together, right? Well, if you had a necklace and you were stringing on these amino acid beads and you went positive, negative, you would find that instead of sitting like a necklace, it would click together. And it would make a little curve right where they all wanted to click together. And you could pull them apart, but as soon as you let go, they would click back together, right? That is the beginning of the way that the side groups of amino acids end up creating the shape or the structure of the molecule that is a protein or a peptide. Let's talk about it a little bit more. Let's talk about polar and hydrophobic, right? Now, anything that is charged is going to be hydrophilic. Anything that is polar but uncharged will also be hydrophilic, right? Hydrophilic means it loves water. Water molecules are put together with polar covalent bonds. You've memorized that by now. And so these guys here in the blue and the purple, they like water. So if I have got a bunch of those in a row and I dip it in water, then those individual side groups would be like, yay, we're in water. And they would just gently float around and be happy. But what if I had a bunch of these guys? All of these guys are hydrophobic. Hydrophobic means they hate water, means they're scared of water. So if I had a bunch of hydrophobic side groups, they would all like click together and form a little ball as they're like trying to hide from the water. Imagine a whole bunch of us that are surrounded by terrifying bats, and we just like glom together with our backs outward, trying to hide from, in this case, the water, right? Now, the interaction of these side chains is what provides them the protein or peptide, its shape, its structure. And I have long been encouraging you to remember that the molecules we're talking about, they actually have real three-dimensional shape. And in the case of proteins, that shape is dictated by the sequence of these 20 amino acids in that particular protein. In a minute, we will talk about how things can go wrong. Now, I want you to imagine that I have got, what was it? Arginine, aspartic acid back and forth. So positive, negative, positive, negative, positive, and they went click. What if I made a mistake? Like it's supposed to click together and have a curve this way. What if I made a mistake when I was putting my beads on the chain and I went, instead of positive, negative, positive, I went positive, 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 negative, positive. Now, they wouldn't click together, right? Because positive and positive push each other apart. So instead of having a bend this way, I actually would have an area that went the opposite way. I would have totally changed the shape of that part of the necklace, that part of our protein. All right. Now, I already showed you this image when I was telling you about how the normal structure of an amino acid is amino with a carbon in the center, hydrogen off there, a carboxyl, and then the side group. Now, the way amino acids get joined together is the organelle called the ribosome that builds proteins. You would have learned that in 150. That organelle, the ribosome, when it's putting together the individual amino acids, it does that by forming a special kind of covalent bond here between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. And that covalent bond is known as a peptide bond. And that is why a short series of amino acids will be called a peptide, and a long sequence of amino acid can be called a polypeptide, but you'll always hear me referring to them as proteins. All righty. So, forming a peptide bond. Here we have got your basic amino acid. The letter R dictates this is a side group. Doesn't tell you which one, just tells you that it is one. There's a little detail here about the side groups going in opposite ways. We don't care about that here in 151. But you can see that the carboxyl group of this amino acid will be attached to the amino group of this amino acid. And in order to do that, this H2O is in the way. And since it's in the way, it needs to leave. So if I were going to take 1, 2, 3, 4 amino acids and put them together to make this very tiny peptide, what kind of reaction is that? Taking smaller things and making them into one larger thing is a synthesis reaction. And since water left, it is called dehydration synthesis, right? And what type of bond was formed? Peptide bonds. This special kind of covalent bond between adjacent amino acids is called a peptide bond. Right? Good. We will start there at the beginning of the next lecture.